In this topic, we'll be exploring a little bit further the effects of different dominance relationships between alleles, building upon what we introduced previously with complete dominance, but also now talking about two different types of dominance relationships. So we said before, complete dominance, dominant allele, always shows its effect in the phenotype. So over here, we have the dominant allele, capital P, which codes for a purple flower. So whether you have two copies of the dominant allele, you're homozygous dominant, or you're heterozygous, either way, you still have the dominant allele. So it shows its effect in the phenotype, and your flowers are purple. This recessive one is being hidden by the dominant, so it doesn't show up in the phenotype. Now the recessive allele, the lowercase p, codes for white flowers. And so homozygous recessive, the only way that shows up is when both copies are recessive. Now you have white flowers. So in this scenario here, what we've done is we've taken a homozygous recessive flower and we've crossed it. That means you've bred it together with a homozygous dominant flower. And then we've used a Punnett square here to show us all the possible combinations of genotypes that can result in the offspring, the children that we made from that combination. As you can see here, all of the offspring will have purple flowers. They will all be heterozygous. Now if you take two of those offspring and you breed them together, one heterozygous offspring bred with another heterozygous, here are the outcomes you get for the individuals from that cross. So the question is, in the second set of breeding plants here, the F2 generation we call it, two questions. What's the percent chance that the offspring would have the same phenotype as the parents? And what's the percent chance the offspring would have the same genotype as the parents? So we're going to take a minute, pause the video, try to work through this, and then we will work on it together and discuss it. So hopefully you've had a chance to do it. If not, go ahead and pause it now. But here we have, for the first part, the phenotype. 3 out of 4, that'd be 75% purple. So that's the phenotype of the parents, so that's be 75% chance, 3 out of 4. And then having the same genotype as the parents. Well, the parents' genotype is heterozygous, big P, small p. There's one of those, and there's another one. So 2 out of 4, 50% chance of having the same genotype. So the reason I did this problem is because it's not particularly difficult. However, you must pay very close attention to these words. Phenotype, genotype. It's going to make all the difference sometimes between having the correct answer and the wrong answer. So make sure you read the problems very carefully on homework and quizzes and tests so you don't mix that up and lose points unnecessarily. Here's an example of a trait controlled by complete dominance, albinism. This is a recessive trait. What that means is that you could have two individuals who are not albinos, but they could produce a child who is an albino. And we'll talk and explore how that can happen later on with a bunch of practice problems, both in lecture and in lab. Albinism is not an exclusively human condition. You'll also find it in many other species. Here we can see a lion, an alligator, a peacock. Here we have a fawn, so a baby deer. Here we've got a snake. Up here you might think this is a squirrel, but it's actually a skunk. It just doesn't have its stripes because it's an albino skunk. And you can even have albino plants. You might wonder, well, how can an albino plant get energy? Because plants need chlorophyll to capture energy from sunlight. Well, that's a very good observation. These are what we call parasitic plants. Unlike most plants that get energy from the sun, these actually feed on nutrients from the roots of other plants in the ground and then take that for themselves. So they're an unusual type of plant. We said that albinism is caused by a recessive allele. It's a recessive trait. There's another condition called progeria. This causes rapid aging. These individuals here are in their 20s and 30s. They look a lot older. Physically, they're a lot older because progeria causes advanced aging. Individuals with progeria do not live usually beyond 30 years old because by 30 their physical age is equivalent to around 90. They are sterile, meaning that they are not able to reproduce, and it's a dominant trait. So if they're sterile, they can't reproduce, and it's a dominant trait, how does this get passed on? That's a great question. It doesn't. It's not possible for progeria to be passed on because they are sterile. However, this being a dominant trait, it means that it shows up due to random mutation. And in the next topic, we'll talk about mutation. But mutation is just a random change in the DNA. And so it's a very, very rare condition. Just this randomly happens by a mistake made in copying the DNA. This leads us to some important points for discussion, though. The fact that this is a dominant trait. A lot of times people think dominant good, recessive bad, or dominant common, recessive rare. Those are not always the case. Progeria, I think there's something like 30 people in the world at any time that have this condition, so it's extremely rare. Most of us would not want to be 90 years old physically by the time we're in our 20s and 30s, so we'd say it's not beneficial. So it's a myth that dominant traits are always more common. It's also a myth that dominant traits are always beneficial. This is not a beneficial trait, even though it's dominant. Similarly, recessive is not always rare. All of us are homozygous recessive for this trait. We do not have progeria. We must be homozygous recessive for it. And then, again, that recessive is bad is not always the case. So now let's introduce a new type of dominance relationship called incomplete dominance. With incomplete dominance, you have two alleles of equal strength. And then when present together, the phenotype is an intermediate blend of the other two homozygous phenotypes. So let's take a look at this. So up here we have complete dominance first for flower color. So we're saying A is the dominant allele coding for red flowers, capital A, and then small a is the recessive allele coding for white flowers. So two of the dominant, or just one of the dominant, still red flowers. The red allele is dominant, shows itself. Now if you have both recessive, then it shows the white. With incomplete dominance, you do not use a capital letter and a small letter, because that denotes one being dominant, one being recessive. Instead, you have two capital letters. With incomplete dominance, one of them you put like a little apostrophe or hash mark next to it, indicating that it's a different allele. So there are two alleles, capital R and capital R with this hash mark next to it. So capital R is an incomplete dominant allele coding for red flowers. R with hash mark, incomplete dominant allele coding for white flowers. So two of the capital R's, you have red flowers. Two of the capital R's with the hash marks, you have white flowers. Now with one of each, this is where that thing I wrote, the phenotype is an intermediate blend. So take red and white paint, for example, and mix them together, you get pink. So it's key that it's intermediate and it's a blend somewhere in between the phenotypes of the other two. It doesn't have to be exactly in the middle, but it's somewhere in that range. That's incomplete dominance. Equal strength, but the resulting phenotype is somewhere in between the other two. It's a blend of those two. Now, codominance is the third and final type of dominance we'll talk about. Unlike incomplete dominance, where the two will blend together, with codominance, they have equal strength but they are separate. So equal strength, they show up separately in the phenotype. That means if you have a capital B and a capital W, capital B codes for black feathers, capital W codes for white feathers. They're equal strength, so they both show up, but instead of blending together and making gray, you have equal but separate. Some black feathers and some white feathers. That's codominance. We'll talk about a few other examples here of codominance. One of those is sickle cell disease, which we go through more in lab. There are two dominant alleles, capital A and a capital S. Two different capital letters indicates codominance. Unlike for incomplete dominance, where it's two of the same capital letters, but one of them had a hash mark next to it. So HBA is short for hemoglobin. Capital A is one of the codominant alleles. HB capital S is the other one. And again, we go through this more in lab. So I'm not going to go through this now, but you can look at this to review from the lab content related to sickle cell disease. You'll want to know it for both lecture and for lab. Similarly, we go through this in detail in lab, but just to have it in both places. For blood types, it's a combination of complete dominance and codominance. Now we have 3 alleles that are present instead of the 2 we've been having in other examples. I with a capital A, I with a capital B. So 2 different capital letters, that's codominance between those 2. Small I means that's recessive compared to either of those. So even though there are these 3 different alleles that affect blood types, each individual just has 2. This is talking about in a population, in a group of people, you might encounter these 3 alleles, but every person just has 2 of those 3. 1 from each parent. So like I said, the 2 different letters that are capital letters are codominant to one another. But if you compare either of those to the small I, they are both completely dominant over the recessive allele. So we have here 6 different genotypes. Genotypes are the combinations of letters. There's 1, 2, 3, 4, 5, 6, right? Those are the genotypes. And over here, the blood types that result, the blood types are the phenotypes. So we have 1, 2, 3, 4 phenotypes that can result from those 6 different genotypes. And the reason for that is that you can have 2 different genotypes for type A, because I with capital A and the small I, that small I is recessive, so it gets hidden by the presence of that dominant allele. Similarly with B, the dominant allele hides that recessive I. The only way you have the I show up, the small I, is if both of them are there, and then you have the type O blood. This is another slide showing the same thing, just in a different way. Here's a quick question, so you can take a minute to pause your video, try to answer this question, and then we'll discuss it in just a second. Okay, so what this question is asking about is you have 2 parents who are heterozygous for a certain trait, but they have different phenotypes. Their child has a phenotype that's intermediate to their phenotype. This is an example of blank. So our key word here is we have an intermediate phenotype. It doesn't show exactly what the parents have. It's some intermediate. It's somewhere in between the 2. That should cue us in then to being incomplete dominance. Remember that was intermediate blended phenotype. So I, incomplete, intermediate, blended phenotype. Here's a good slide that summarizes these 3 relationships, their effects, and some example letters that could be used to represent those is an easy way to try to study this for when you're working on preparing for tests. And that finishes up that topic.